Is it the same one?
Can you go on and check? Fantastic. They got confused with the sign. It's on. Nice. All right, I think I have the sound working now. Is it good? Great, thank you. That was my producer, Lulu. So I think all the audio-visual stuff is fine. We're streaming, everything's okay. So thank you everyone for coming into my public defense. I'm David Ford, a PhD student at Loyola University Chicago on the Health Sciences campus. I'm in the Biochemistry and Molecular Biology program, and I've been in the lab of Andrew Dingwall for 6 years now. Well, going on about 7, since 2013 is when I joined Loyola. And so I'll be presenting to you a summation, abbreviated summation of my research today. I'd like to say that there is a slight delay in this recording. So what you'll be seeing is me speaking about 30 seconds to a minute delayed. Please feel free, if you have any questions throughout the presentation, to type them into the chat. And then at the end of the presentation, there will be time for me to go through them. Okay, so I'm going to go ahead and begin. So, you've all been looking at this. This is the development of the newt from a single cell. And just like the newt, we all begin from a single cell that through multiplication becomes trillions of cells that are individually formed in form and function in order to create a living and working animal. Now, this developmental process involves multiple stages of differentiation. Differentiation is when a relatively undifferentiated cell, which has the capability to become many different cell types, then adopts its fate and changes its form and function accordingly. Now, of course, all cells undifferentiated and differentiating within the same organism, all of our cells, have the exact same genome, the same DNA, the same genes. And in order to change during differentiation, they must change their gene expression, which genes are on and which are off. Now, this process is called reprogramming, and it's absolutely critical for development and any other changes in gene expression. Now, when we think about changes in gene expression, we think about the promoter region, because the promoter is where the coding sequence of a gene starts. And of course, we know that when transcription factors bind to the promoter region, they have activating ability and promote transcription. However, during reprogramming, just simple binding of transcription factors to their targets usually only elicits very weak transcription. That's spotty and stalling. In order for full, efficient activation during reprogramming, these require enhancer regions. Enhancers are sections of DNA that do not code for protein, but rather their function is to regulate other sections of DNA that do. In fact, it is the complicated pattern of enhancer activation in combination with promoters, with their target promoters, that allows the complex multicellular development that leads to organic life. So that is how promoters are activated, how genes are transcribed after reprogramming, but how are enhancers controlled? Well, to talk about that, we have to talk about chromatin. Now, the DNA in our cells is not loose in the nuclei. Instead, it's packaged into a nucleic acid and protein composite known as chromatin. Now, chromatin not only organizes the DNA, but also is necessary for marking it and controlling its activity state. For example, this is an example of an enhancer, specifically a repressed enhancer before it is activated for reprogramming. As you can see here, it is very compact. It's not in contact with any promoter region, and it's decorated with this modification, H3K27 trimethylation. This is a chemical group added to the chromatin, to the histone proteins, that is associated with repression. In order to activate this enhancer region, pioneer transcription factors bind to it, and then they recruit a number of chromatin remodeling machinery. This loosens the chromatin environment and allows the recruitment of other factors. This includes MLR complexes. MLR complexes are a type of compass-like complex, which means they are epigenetic regulators that function through histone modification. But all you need to know about them right now is that they are the necessary tools that are brought in order to activate enhancer regions. They have a number of different functions. These include the deposition of a new tag, H3K4 monomethylation, which is associated with activity of enhancers. These also carry the function to remove the repressive trimethylation of K27 and recruit the factors that replace it with activity-associated acetylation. Then this enhancer is fully primed for activation, fully established. It can then come in contact with large complexes of proteins, including mediator and polymerases, which then bring it in contact with its target promoter regions and allow for efficient transcription of the targets and complete reprogramming. Now, MLR complexes are fairly promiscuous tools. They interact with a lot of different transcription factors which use them to activate their target enhancers. It's known that they interact with a number of different lineage-determining factors, which are the transcription factors necessary for cell fate choice during differentiation, such as if a cell is going to be a macrophage or a neuron or a muscle cell or what have you. They're also required for nuclear receptors, such as hormone receptors, or others that respond to outside stimuli of the cells. Very importantly, they interact with and are necessary for regulating the targets of a number of different highly conserved developmental signaling pathways. This includes hipposignaling, TGF-beta, and Notch, to name a few. This means that they use the MLR complex in order to fully reprogram the cell during organ formation, allowing for organ formation and correct tissue patterning. Now, humans, we have two MLR complexes that have two central methyltransferase proteins. These proteins are absolutely necessary for the formation and the function of our MLR complexes. They're known as KMT2C and KMT2D. Now, it's known that the genes encoding these proteins in humans are two of the most frequently mutated genes in almost every form of human cancer. As you can see here, KMT2C and D are mutated to a frequency of 20 to 50% in almost every solid tumor type, significantly high. In addition, germline mutation of KMT2C and D is associated with developmental disorders in humans. These mean that alteration of MLR complex function in humans is foundational to disease states. But unfortunately, we don't yet know exactly how this leads to the disease, and therefore we can't properly target it therapeutically. In order to understand that, we need model organisms, such as the fruit fly Drosophila melanogaster. Now, because MLR complexes are ancient and so highly conserved, Drosophila has an MLR complex that's nearly identical to the one in humans. It contains two central proteins, CMI and TRR, which through a likely ancestral genetic splitting event correlates to the N-terminal half and the C-terminal halves of KMT2C and D in humans. However, CMI and TRR come together to form a virtually identical complex with conserved form, function, interacting partners, and regulatory targets. This allows me to use Drosophila as a developmental model to better understand how the MLR complex works. Allow me to elucidate the targets and functions of this complex during development of the fly. Now, most of my work involves modulating the levels of CMI and TRR in specific tissues in the fly as it develops, tracking the phenotypes, which is the physical changes that result, and then identifying the molecular causes. In order to do this, I've used the GAL4 expression system. Now, put very simply, what this system allows me to do is increase or decrease the amount of any protein of my choice in virtually any fly tissue of my choice at any developmental time point. It's an extremely useful tool. And put simply, it operates like this. It's a combination of a driver and a target. The GAL4 driver expresses this transcription factor, the GAL4 transcription factor, according to fly genomic enhancers and regulatory sequences. This allows me to choose exactly in what tissue this transcription factor is expressed. And then its target is activated by GAL4 in whatever that tissue is. And the target can be whatever gene product I choose. So I could overexpress, ectopically express, or even knock down targets of my choice. So mostly this is used for increasing and decreasing CMI and TR levels in order to modulate the activity of the MLR complex. And the first story that I'm going to tell you today is how I use this system in order to study how the MLR complex regulates the development and survival of the Drosophila eye. So this is a compound eye of the fruit fly. It's made up of between 700 and 750 individual ommatidia, which are the individual compound units of the eye. As you can see, in a healthy, normal, wild-type eye, this forms a very large surface that's extremely smooth and precisely patterned. Now, we found that when we knock down CMI or TRR in the eye as it's developing, it leads to this phenotype, a range of rough and shrunken effects from the very slightly affected to the completely rough and shrunken, and in some cases, those missing or having malformed organs. In fact, there is a slight severity discrepancy between CMI and TRR knockdown. When I reduce CMI in the developing eye, it leads to an effect of incomplete penetrance, where approximately 1/4 of the animals remain wild type, and the rest have a ranging phenotype of the slightly to completely rough and shrunken. When I knock down TRR, this leads to a more severe version of this phenotype. All of these animals have completely rough and shrunken eyes, and a significant portion of them have missing or malformed organs. Now, this roughness I've discovered is due to mispatterning of the ommatidia, as you can see here through electron microscopy images, as opposed to the normal wild-type eye, which is precisely and perfectly patterned. You can see the hexagonal structure. Every single ommatidial unit borders exactly 6 others and borders exactly 3 bristles or little hairs in the eye. But when I knock down CMI or TRR, this leads to a disruption of that patterning. It changes the function and size of the borders of this lattice. It changes the patterning of the bristles. Some are lost, some are duplicated, and in some extreme cases, complete lens fusion of the neighboring ommatidia. This data demonstrates that the MLR complex is required for proper eye development. However, it does not yet explain what causes this rough and shrunken phenotype. In order to investigate this, I have to turn to tissue known as the Drosophila eye disc. The eye disc is an imaginal disc, which means it's the progenitor tissue in the larva that will develop into adult organs during metamorphosis. Now this, as you can clearly see, the eye disc develops into the adult antenna and the adult eye, which is clearly separated into 2 individual lobes. I, of course, am more interested in the eye pouch. Now, as you can see here, assuming that the resolution on your computer screens is good enough, the eye pouch has a very interesting aspect to it, and that is an indentation that runs across the middle. This is known as the morphogenetic furrow, and it's actually a mobile boundary that moves from the posterior margin of the eye to the anterior as the organ develops. And this induces differentiation of the eye cells as it passes, meaning that all the cells anterior to the morphogenetic furrow remain undifferentiated, whereas those posterior to the furrow are currently reprogramming and differentiating into adult eye tissue. Now, this is very useful because it allows me to look at one piece of tissue, to look at eye cells at different states of development. Now here is another example of an eye disc. In blue, I've stained for DAPI, which stains nuclei so that you can see the outline of the disc. In red is ELAV. ELAV is expressed in the differentiating cells. This allows me to distinguish the undifferentiated from the differentiating tissue within the same organ. In green stands for cleaved DCP1. DCP1 is an effector caspase in Drosophila. Cleaved DCP1 demonstrates caspase cascade and suggests apoptotic cell death. A clear and obvious explanation for the rough and shrunken organs that I found upon CMI or TRR knockdown is aberrant cell death during development, causing organ shrinkage and disrupting the patterning. When I knocked down CMI or TRR in the eye disc, I saw this effect. There was a clear increase in caspase activation only within the undifferentiated tissue in CMI and TRR knockdown discs, and most of this was concentrated in a very specific region, right on the dorsal ventral boundary in the middle. In order to verify that this caspase activation is actually leading to apoptotic cell death, I used tunnel staining, which is a classical assay of apoptosis. This tunnel staining demonstrated that I found fragmented DNA, which is a sign of apoptosis, within these same cells where I am seeing DCP1 activation, confirming apoptotic cell death. Now, this cell death could be due to the fact that these cells are lacking the MLR complex and it's necessary to protect them. Or perhaps neighboring cells lack the MLR complex, are not giving them the signals that they require to survive. To determine if the effect is cell autonomous or non-autonomous, I took advantage of the Gal4 system again. And I was able to knock down CMI or TRR only within the dorsal half of the organ, leaving the ventral half completely wild type as an internal control. This demonstrated that the DCP1 activation, the caspase cascade, occurred only within those cells that were lacking CMI or TRR, demonstrating that the MLR complex has protective function in the undifferentiated eye tissue against apoptotic death. Now, this could be unique to the eye, or it could be shared among different imaginal tissue that's undifferentiated. In order to investigate that, I turned to another imaginal disc, the wing disc. As you can imagine, the wing disc develops into the adult wing. Using the GAL4 system, I knocked down either CMI or TRR only within the posterior margin of the wing, leaving the ventral portion wild type. In red is DCP1 activation again, as you can see from the merged image. DCP1 is significantly activated. It's clearly induced in those tissue that is lacking CMI or TRR, further demonstrating that the MLR complex has a general protective role against apoptosis in undifferentiated imaginal tissue. The question is, what is the mechanism of this protection? I think my mic was changed for a second, but I've addressed that. So, what is the mechanism of this protection? As the MLR complex is known as a gene regulator, it's likely that something dysregulated in these cells is causing cell death. And a likely candidate for that is the microRNA bantam. Now, microRNAs do not code for protein as mRNAs do. Instead, they function by inhibiting the translation of their target mRNAs. Therefore, they are a negative regulator of gene expression. Now, the best characterized function of the bantam microRNA is the inhibition, the suppression of apoptotic cell death during development in the undifferentiated imaginal tissue. In addition to that, expression of bantam is regulated by a number of highly conserved developmental signaling pathways. This includes Hippo, DPP, which is a TGF-beta, and Notch signaling. And if you were paying attention earlier, you'll know that the MLR complex interacts with all of these pathways and is necessary for regulating their transcriptional targets. Therefore, and it's quite easy to assume that the MLR complex is also required for regulating the expression of the bantam microRNA. And if so, when the complex is knocked down, there could be less expression of bantam microRNA, leading to this cell death. Now, we know that the MLR complex regulates enhancer regions. So if this is true, that means that the MLR complex localizes to bantam-specific enhancers. It regulates the activity of those enhancers, which then leads to control of bantam transcription. This is ChIP-seq data prepared by Claudia in our lab, and it demonstrates CMI binding throughout the genome. This is just a window of the bantam encoding sequence and the local regulatory locus at different stages of development, in the embryo, in the late larva, and within the early pupa. Now, even though bantam is encoded all the way over here, these two are previously identified regulatory sequences for bantam, the bantam wing enhancer and the bantam eye enhancer, which has been shown to replicate bantam expression in the wing and the eye disc. As you can see here, CMI is enriched at peaks at these enhancers in the late larva and early pupa, the time points at which the imaginal discs are developing and bantam is being regulated in them. This is very suggestive that the MLR complex has regulatory activity at these enhancer regions. But in order to demonstrate that, I've taken advantage of reporter constructs. These reporter constructs use beta-gal expression as a readout of the activity level of these enhancers. So this is an example of the activity pattern of the bantam wing enhancer and where it is active within the developing wing disc. Now, I would imagine if the MLR complex is required for promoting bantam expression and positively regulating its enhancers, then when I knock down CMI or TRR, this would lead to a decrease in the level of activation. However, I found the exact opposite. CMI or TRR knockdown leads to a significant increase in the activity of the bantam wing enhancer, suggesting the complex serves to suppress its activity. Turning to the eye, this bantam eye enhancer, where the complex localizes to, is active in a very strong and stable pattern at the anterior margin of the eye disc. When I knock down CMI or TRR, I don't see a clear increase or decrease in the bantam eye enhancer activity. Instead, I see that there is a disruption of the patterning, suggesting that the MLR complex doesn't function to either promote or suppress it, but may function in order to establish the spatiotemporal activation of this enhancer region. Now, altogether, this data demonstrates that the MLR complex directly regulates the activity of these tissue-specific bantam enhancer regions, which is very suggestive, but I haven't yet demonstrated that this leads to regulation of the bantam microRNA itself. So first, I'm going to turn to the wing. Now, this is qPCR data prepared by Claudia in our lab, and it demonstrates that bantam is significantly increased in the wing discs upon knockdown of either CMI or TLR, agreeing with what I just showed you of suppression of the bantam wing enhancer by the MLR complex. In order to verify this and visualize it in the tissue, I took advantage of this tool, the bantam sensor. The bantam sensor acts as an inverse reporter of bantam activity using GFP as an inverse readout. Put simply, the higher the level of bantam in a cell, the lower the level of GFP expression, and vice versa. Therefore, an inverse sensor. Here's an example of it in the wing. When I knock down CMI or TLR only within the posterior margin on this right side of the wing disc, compared to control, both of these knockdowns demonstrated significantly lower GFP expression, meaning significantly higher bantam activity. All of this data agrees and suggests the MLR complex functions in the wing disc to suppress bantam expression. However, my next question is whether or not this has developmental consequence, and so I turned to the adult wing. Now, our lab has previously characterized wing phenotypes associated with either CMI knockdown or overexpression. Compared to the normal wild-type wing with its precise wing vein patterning, when CMI is knocked down as it develops, it causes slight vein retraction. And when CMI is overexpressed, it causes slight ectopic vein formation, vein end forking, and a reduction in organ size. Now, I'm able to modulate the activity of bantam in the background of CMI knockdown or overexpression. The resulting phenotypes help inform me whether or not the bantam microRNA and the MLR complex interact phenotypically. When I reduce bantam activity through a microRNA sponge in the background of CMI knockdown, this completely suppresses the wing vein retraction phenotype, completely rescuing the wild-type wing, suggesting that the microRNA bantam acts antagonistically towards the MLR complex activity. This is verified because when the bantam sponge is expressed in the background of CMI overexpression, this causes a significant enhancement of the wing vein phenotypes, more ectopic veins and more vein splitting. I found the reciprocal is also true. When I increase bantam activity through overexpression, this leads to an enhancement, a significant enhancement of the wing vein retraction phenotype found in CMI knockdown wings. And although overexpression of bantam does not completely suppress the wing vein phenotypes in the CMI overexpressing background, it does rescue wild-type wing size, further confirming that the MLR complex and the bantam microRNA act antagonistically towards each other phenotypically, and suggesting that it's very critical for the MLR complex to suppress bantam expression during organ formation. Which leads me to this model. Now, it's been previously determined that DPP signaling, which is a form of TGF-beta signaling, is required in the wing disc for proper organ formation, both the size and the vein patterning. Previously, our lab has determined that the MLR complex is required for DPP expression in the wing disc, allowing for proper vein formation. And recently, another group has determined that the bantam microRNA actually acts as an inhibitor of DPP signaling activity during wing development. The results I show you today paint this picture, which is that the MLR complex promotes DPP activity and allows for proper wing development at two levels in the wing, directly through the promotion of DPP expression itself, and indirectly through suppressing the expression of inhibitor bantam. But that is the wing, and I started with the eye. So I'm actually able to do very similar interaction experiments in the eye as I performed in the wing, by modulating the activity of bantam in the background of CMI or TRR knockdown, tracking the phenotypic changes which result in the population, and then using that to interpret whether or not they interact phenotypically with each other. Now, in the background of CMI or TRR knockdown, increased bantam activity through overexpression causes a significant enhancement, a worsening of the rough and shrunken eye phenotype. Reduction of bantam activity through the sponge suppresses this rough and shrunken phenotype, suggesting that in the eye, just as I saw in the wing, bantam and the MLR complex are antagonistic towards each other. Importantly, bantam overexpression by itself without affecting the MLR complex at all phenocopies mimics the slightly rough and shrunken phenotype, suggesting that perhaps alteration, dysregulation of bantam expression, is what's causing this effect. I found that the same is true if I look at the ommatidial level. In the background of CMI or TRR knockdown, bantam overexpression matches or enhances the patterning defects found in the compound eye, whereas reduction of bantam activity actually rescues and slightly suppresses these mispatterning defects. And critically, bantam overexpression by itself shows the exact same lattice disruptions and patterning defects found in CMI and TRR knockdown. So, I've demonstrated that they interact phenotypically, and earlier I showed you that the MLR complex is required for the correct regulation of a bantam eye enhancer. But I have to demonstrate that the MLR complex actually is important for regulating bantam transcription itself. As I explained to you before, the eye disc is a complicated organ. It's a tissue that contains both undifferentiated and differentiating cells separated by the morphogenetic furrow, which I've marked with blue arrows here. And as it turns out, bantam expression is different in these different cell populations. As you can see here with the bantam sensor, there is relatively low GFP expression in the undifferentiated cells, meaning high bantam activity. Whereas as they begin differentiating, GFP goes up, which means bantam is downregulated. Now, in order to best investigate this complicated tissue and how the MLR complex may affect bantam expression within it, I took full advantage of the GAL4 system. Now, all of the data that I showed you before with the eye, all of the phenotypes, the rough and shrunken, the cell death, that was all performed using this eyeless GAL4 driver. As you can see here, through staining of CMI, I'm able to knock down CMI or TRR within the entire eye pouch, both the undifferentiated and the differentiating cells, leaving the antennal section wild type. I'm also able to use the GAL-B69B driver, which is ubiquitous and allows me to knock down within every single cell in this tissue in order to verify. I've already showed you the dorsal eye GAL4 driver, which allows me to knock down only within one half of the disc of the dorsal, leaving the ventral half wild type as an internal control. And finally, I have the GMR GAL4 driver. GMR allows me to express knockdown only within these differentiating cells at the posterior, leaving the anterior undifferentiated cells with wild type CMI and TRR levels. All of these used in conjunction with the bantam sensor will allow me to properly investigate how the MLR complex regulates bantam expression within the developing eye. So first, as you can see here, as I hope you can see, depending on how good the resolution on your computer screen is that in my control discs, of course, bantam is relatively high in the undifferentiated tissue and relatively low in the differentiated tissue, as can be seen by this high GFP level here, which I've magnified in these little insets. Now, when I knock down CMI or TRR with the eyeless GAL4 driver in both the undifferentiated and differentiating cells, I have two observations. The first and the most obvious is the fact that there's a significant increase in GFP expression within these differentiating cells, meaning bantam is downregulated upon loss of MLR complex activity. In the undifferentiated cells, there's absolutely no difference in bantam activity compared to my control discs, meaning that the MLR complex is unnecessary in that tissue in order to regulate bantam expression. I confirm this using the GAL-B69B GAL4 driver, which exactly matches what I saw with eyeless. When MLR complex is lost within every single cell in this tissue, I see the no change of bantam expression within the undifferentiated cells, but a clear decrease in bantam expression as seen by higher GFP in the differentiating cells. This is even clearer by using dorsal eye GAL4, where I can compare my knockdown dorsal to my wild-type ventral, showing the clear difference in bantam regulation within these differentiated cells. Now, all of this data suggests that the MLR complex is unnecessary in the undifferentiated cells for regulating bantam expression, but it is required within the differentiating cells. If that's true, when I knock down CMI or TRR with this GMR-GAL4 driver only within those differentiating cells, I would expect to see the same increase in GFP activity from the sensor. However, I see the opposite. Instead, these tissues exactly match bantam expression to the control. This means that the MLR complex is not necessary within the differentiating cells in order to properly regulate bantam expression. Now, the only difference, you can see it quantified here, but the only difference between this driver and these three is that these three all also knock down CMI or TRR within the undifferentiated tissue. This means that the MLR complex has necessary regulatory activity within the undifferentiated tissue, likely the establishment of enhancers, that is required for proper bantam regulation cell generations later, once differentiation commences. Now, this result actually mirrors what has recently been shown within mammalian cells through in vitro experiments. Other groups have demonstrated that MLR complex activity is required for reprogramming, but once that reprogramming is complete or before it's done, the MLR complex is unnecessary, it's dispensable for maintaining that gene transcription. Now, I have not only verified this in an in vivo developmental model, my results have gone further to demonstrate that the MLR complex has establishment activity, necessary regulatory activity in undifferentiated cells, in some cases, cell generations before the actual differentiation event, giving us valuable data into the timing of enhancer establishment during development. Now, before I move away from this, something important to keep in mind is that bantam is being dysregulated in these differentiating cells. These cells are taking on new cell fates. They're adopting different lineages. And I wanted to investigate whether all of these different lineages are similarly sensitive to the MLR complex and required for bantam regulation. Now, one of the first fate choices after the morphogenetic furrow passes and differentiation begins within the eye is whether to adopt the proneuronal lineage or the interommatidial lineage. Proneuronal cells will develop into the photoreceptors and the cone cells, all the neural tissue of the eye, whereas the interommatidial cells will develop into the pigment cells, bristle cells, and just generally the infrastructure making up the compound eye lattice. Fortunately, I can easily distinguish these two cell lineages. ELAV, which I demonstrated to you before, marks the differentiating cells, is only expressed within this proneuronal lineage, shown here in red, meaning that these negative cells, the black cells, are of the interommatidial lineage. As you can see here, when I co-stain for both ELAV and the bantam sensor GFP, I see a clear localization of the ELAV and GFP, meaning that these proneuronal cells have relatively low bantam expression, whereas the surrounding interommatidial cells have relatively high bantam expression. Now, when I knock down CMI or TRR, I see two effects. The first and most obvious of which is all of this GFP activity, which I showed you on the earlier slide. All of that only occurs within these interommatidial cells surrounding the developing ommatidia, meaning that upon CMI or TRR loss, this causes a significant downregulation of bantam within these cells and suggests the complex is important, is required to positively regulate bantam in this lineage. A little more subtle is the second effect, and that is that the bantam sensor decreases within these proneuronal cells at the center of the ommatidia, meaning that bantam expression is upregulated in these cells upon the loss of CMI or TRR. So I've demonstrated that the complex is required to positively regulate bantam within the interommatidial lineage, and simultaneously it negatively regulates bantam within the proneuronal lineage. This is the first example of an MLR complex regulating a single transcriptional target in opposite directions, depending on the context of cell fate, a very critical developmental regulatory tool. Which leads me to this model of bantam expression and regulation in the eye. Within the undifferentiated eye disc, bantam is highly expressed. Upon reprogramming and differentiation, within the proneuronal lineage, bantam is downregulated, whereas in the interommatidial lineage, bantam is significantly upregulated. It's kept high. I've demonstrated that the MLR complex has necessary regulatory activity and enhancers within the undifferentiated eye disc that does not, at that time, affect bantam expression. However, that activity, that likely establishment, is necessary later on to both suppress bantam expression within the proneuronal phase and promote bantam expression within the interommatidial phase. Now, if you were paying attention, which I understand I'm throwing a lot at you, but if you were paying attention, you can see there's a problem. And the problem is, the whole reason I was looking into regulation of bantam is to try to explain exactly what's causing this cell death phenotype upon CMI or TRR knockdown in order to explain the rough and shrunken phenotype. Now, as I demonstrated to you, bantam dysregulation upon CMI or TRR knockdown only occurs within these differentiating cells on the posterior, whereas I showed you that the apoptosis, the caspase induction, only occurs within the undifferentiated cells on the anterior. They're mutually exclusive events, and therefore they cannot be causal. This cell death is not due to dysregulation of bantam. Now, even though it is not due to dysregulation of bantam, it still could be sensitive to it. And I've demonstrated that when I overexpress bantam in the background of CMI or TRR knockdown, that causes a significant enhancement of the caspase activation phenotype. And then when I reduce bantam activity, that causes a suppression of that phenotype. Now, this agrees with the data that I showed you of bantam interaction in the rough and shrunken eye. I have two phenotypes, the cell death phenotype in the wing disc and the rough and shrunken eye in the adult. Both of them are enhanced by increased bantam expression, and both of them are suppressed by decreased bantam activity. This is very suggestive that the two are mechanistically linked. But in order to verify that, I used P35. P35 is a pan-caspase inhibitor. It's very good at suppressing caspase cascade and preventing apoptotic cell death. And as you can see here, it works. When I express it in the background of CMI or TRR knockdown, this successfully suppresses the caspase activation phenotype associated with the knockdowns. Now, if this increased cell death leads to the rough and shrunken organs of the adult eye, I would expect that P35 expression would also suppress that phenotype. But, as it turns out, the opposite is true. The rough and shrunken phenotype is significantly enhanced by the expression of P35. And P35 overexpression on its own is actually able to phenocopy, to mimic the rough and shrunken phenotype. Now, this leaves me with a lot of questions, but it also provides answers. Namely, this demonstrates that the two phenotypes, the apoptotic cell death within the eye disc and the rough and shrunken eye in the adult, are not mechanistically linked. In fact, they are completely separate phenotypes that are not causal to each other, different effects caused by the loss of MLR complex activity. This also demonstrates both of those phenotypes, even though they may not be caused by bantam dysregulation, they are sensitive to it. And that is a very suggestive clue. That suggests that whatever is dysregulated causing these effects is sensitive to bantam levels, meaning it likely involves a regulatory target of bantam. And I apologize that I can't get into the details of it right now due to time constraints, but I do want to explain my hypothesis. And my hypothesis is that it is dysregulated Notch signaling, which is leading to both the apoptotic cell death and the rough and shrunken phenotype. Notch signaling in the eye is partially regulated by bantam expression, as bantam inhibits the translation of Notch inhibitor num, thereby promoting Notch activity. And the MLR complex has been demonstrated in multiple organisms to either repress or promote Notch signaling activity. It's known that in the eye, Notch is active in this very specific region, right on the dorsal ventral boundary down the middle of the eye disc. it has functioned in this region in order to promote survival of the undifferentiated cells. And as you remember, when I knock down CMI or TRR, this leads to apoptotic cell death in this exact same region, suggesting perhaps these are dying because there is not proper Notch signaling there to promote their survival. In order to investigate this, I use a Notch response element GFP reporter in order to determine how the Notch targets are activated or inactivated based on CMI or TRR knockdown. I demonstrated that when CMI or TRR are depleted, this leads to a significant decrease in Notch response element activity, suggesting that the MLR complex is required to activate Notch targets in the eye disc, and agreeing that it could be due to the loss of Notch signaling activity causing cell death in this region. So I also turned to the eye to see if this relationship was conserved. I was able to increase or decrease Notch signaling activity in the background of CMI or TRR knockdown. And I understand a lot is going on in this slide, and I'll run through it quickly, but I want to give you the gist. When I decrease Notch signaling activity in the background of CMI or TRR knockdown, this suppresses the rough and shrunken phenotype. When I increase Notch signaling activity, it significantly enhances and worsens the rough and shrunken phenotype. And critically, Notch signaling activity induced by itself increased ectopically high activity in the absence of CMI or TRR knockdown phenocopies the rough and shrunken eyes. This data is very suggestive not only that in the adult eye, the MLR complex and Notch signaling are antagonistic towards each other, but also that ectopically aberrantly high Notch signaling activity could be causal to the rough and shrunken adult eye. So here is my current hypothetical model. That in the undifferentiated eye disc, the MLR complex functions to promote Notch signaling activity and is required for activation of its target genes. This allows for its promotion of cell survival, and when the MLR complex is lost, the cells die. Later on in development, in the differentiating compound eye, aberrantly high Notch signal activity I showed you results in the rough and shrunken adult eye, and therefore I believe the MLR complex has a role in suppressing this activity to promote proper eye formation. Now, I've been speaking for a while, so I'm going to take a drink, but I have one more story that I would love to share with you. Unfortunately, this is going to be a much shorter story than the first one. Due to time restraints, I am going to rush through this a little bit, and it's unfortunate that I won't be able to give you the details, but I think it's important to deliver the high points. Now, this investigation was inspired by data in mice, completely separate from all my previous work. And this data in mice demonstrated that loss of MLR complex activity during mouse development significantly affects the adipose, the fat tissue, and the liver tissue, causing inability to differentiate and form, and also affecting gene transcription. Now, I wanted to see whether this was conserved within the fly so I could better understand how this happens. The fly actually contains an organ known as the fat body, which you can see here as I visualize at different developmental stages. The fat body is actually analogous to both mammalian fat and liver tissue. It's the main storage site of triglycerides as fat, and it's a critical hormone, metabolic, and stress response organ. During the life cycle of Drosophila, most of the early stages, the larval stages, are involved with the storage of fat and gaining more fat as energy. This fat is then used to power metamorphosis in the pupal stage and also remains in the adult in order to give them energy at times of starvation or nutrient deprivation. The work that I'm going to show you right now involves modulating the levels of CMI or TRR specifically within the fat body, and then tracking to see how this affects the animal's ability to survive nutrient stress states such as metamorphosis or starvation. Now, first, in the pupa during metamorphosis, I discovered that although CMI knockdown did not affect this animal's ability to develop or survive, CMI overexpression within the fat body sensitized it to death at higher metabolically stressful temperatures, resulting in animals that were completely formed adults that did not exit the pupa and died inside. This data was very suggestive that it's not due to any developmental deformities, but rather the animals likely ran out of energy and died. During adult starvation, I determined that here in yellow, CMI knockdown animals were CMI knockdown within the fat body were able to survive significantly longer than control and CMI overexpressing animals. Now I turned to see how they deplete their fat, how they deplete their stored triglycerides during both these periods. As you can see here, after metamorphosis, CMI overexpressing animals had significantly less leftover stored fat compared to control and knockdown, demonstrating they burn through it faster when CMI is overexpressed, sometimes leading to death. Whereas over here, the CMI knockdown animals, which were able to survive significantly longer during starvation, actually deplete their fat at a significantly slower rate, explaining that effect. This suggests that somehow the MLR complex in the fat body is required for promoting triglyceride depletion during these nutrient stress states. But as it is a gene regulator, the question is always, what are the dysregulated genes that are causing this effect? And for that, we turn to RNA sequencing. We sequence the transcriptome of the larval fat body in order to determine how gene expression is affected by increase or decrease of CMI level. And unexpectedly, we did not find any significant changes in possible candidate metabolic genes or metabolic pathways or general fat metabolism groups. What we did find is that significantly affected by CMI knockdown or overexpression were stress signaling expression pathways. This includes antimicrobial stress. This includes innate immune, heat, oxidative stress. All different types were all dysregulated upon modulation of CMI, suggesting that the complex is critical for regulating this reprogramming during stress response. And a transcription factor that is critical for orchestrating that reprogramming is FOXO. FOXO is highly conserved. It's activated during stress states, and then it reprograms cells in order to survive those stress states. Now, I found that a number of classic FOXO transcriptional targets within the fat body were dysregulated upon CMI knockdown or overexpression. CMI knockdown led to a significant upregulation of these, and CMI overexpression led to a significant downregulation of all of these. Now, this suggests that in the larval fat body, the MLR complex suppresses FOXO activity during nutrient stress. Now, I sought to see if it does the same within the adult, and if that could explain the metabolic phenotypes that I observe. Now, unexpectedly, again, I received the opposite result. Instead of antagonistic or inverse to each other, FOXO activity and MLR complex activity were directly correlative. FOXO knockdown in red, the same as CMI knockdown, provided a similar protection against adult starvation. And CMI knockdown in yellow provides a similar protection as FOXO knockdown to oxidative stress survival. Now, this suggests that perhaps the MLR complex actually promotes FOXO transcriptional activity within the adult fat body. Which leads me to this hypothetical model. One of FOXO targets within the fat body during nutrient stress is the lipase bremmer. Bremmer functions to increase the lipolysis depletion rate of the stored triglycerides, meaning that if the MLR complex is responsible for promoting FOXO activity in the fat body during nutrient stress, CMI upregulation, CMI overexpression would cause increased TAG lipolysis and lead to the metamorphic death, whereas CMI knockdown will lead to decreased lipolysis and allow these animals to deplete their fat slower and survive longer. Now, in addition to all of the results and conclusions that I've explained, I also want to leave you with the main impacts from my work. This includes the fact that the MLR complex is required within undifferentiated tissue for enhancer establishment and proper transcriptional regulation after differentiation. Now, this is for the first time demonstrated in an in vivo developmental model and provides new evidence elucidating how these enhancers are established and exactly what the timing is during organism development. I've also demonstrated that the MLR complex is required in undifferentiated tissue to protect against apoptotic cell death, and suggested that perhaps dysregulated NOT signaling may be involved. Usually when we're talking about differentiated cells, the MLR complex is associated with being required for reprogramming. But I've demonstrated using my developmental model that it also is required for survival before that reprogramming. I've also shown that the MLR complex is required to regulate stress transcription in the fat tissue. Now, this is an understudied area, reprogramming in a terminally differentiated cell type. It shows the MLR complex is also used for this critical survival reprogramming event. And finally, and I think most importantly, I've demonstrated the MLR complex may either promote or suppress, either positively or negatively regulate its targets or the activity of its partner transcription factors, depending on the contexts of development, differentiation, and cell fate determination. I've demonstrated this with differential regulation of bantam, and suggested the same with different regulation of Notch signaling activity and different regulation of FOXO activity within different developmental stages. Now, this further elucidates and adds a new dimension to exactly how the MLR complex functions. The final thing I want to leave you with is a quick discussion of negative regulation by the MLR complex. As I explained at the very beginning of this presentation, the MLR complex is known as a tool that is brought in to activate, to positively regulate target enhancers. And negative regulation of enhancers and negative regulation of gene transcription by the complex is relatively uncharted territory. I say relatively because just a few months ago, our lab published a new paper providing a new paradigm to exactly how the MLR complex may function. And we demonstrated that the MLR complex is actually required at certain targets in order to suppress them to prevent premature activation before the appropriate reprogramming time point. And of course, during that reprogramming, that establishing enhancer is necessary for activation of the genes. Now, this means that when the MLR complex is lost, it has two effects. First of which, some of its targets are activated prematurely before the normal reprogramming step. And then once that reprogramming step occurs, these targets fail to activate. Now, these results, in combination with my further examples of how this works during development, in stress states, and metabolism with various developmental signaling pathways, further elucidate exactly how the MLR complex functions. And better understanding of that and these new dimensions to its regulatory activity that were not previously assumed will give us a clearer window into exactly what's going wrong with these human disease states. So, thank you all for listening to this presentation. It's been very strange delivering this defense from my apartment wall in the middle of the coronavirus quarantine, I guess you could call it. But as much as the people aren't here today, I would love to thank them for everything they've done for me during my process as a graduate student. It may sound like I'm getting emotional, but my throat's very raspy from speaking. But I, of course, want to thank all the members of my lab, specifically Dr. Andrew Dingwall, who is my mentor, who always has acted less like a boss and more like a true mentor, with patience, with understanding, with clear training of how to become a scientist, with a focus always on growing my career and helping what I need to do to advance as a scientist and as a person. So I want to thank him for all of that. I would not be who I am or where I am today without his guidance. I also would like to thank Claudia in our lab. Claudia is the senior researcher in our lab, and I cannot count the number of times that she's helped educate me in various assays or procedures or different experimental techniques. She's shared her wealth of knowledge and experience with me and allowed me to develop as a scientist so much. And in addition to all of that technical expertise, she's always been able to help speak on theories or hypotheses or where my research should go based on that. And so I can't thank her enough for everything that she shared. I'd like to thank the other members of my lab. Tim Nichols, who was a master's student while I was there, who has since gone on to bigger and better things, is doing very well right now. I'd like to thank Abdul Zakar and Jeffrey Ng, who were the bioinformatics students who helped me deal with some of this high throughput data, the ChIP-seq, the RNA-seq, all of this stuff. They helped us get actual working data out of it. My dissertation committee, the head of which is Dr. Mitch Denny, in addition, Dr. Nancy Zalesnik-Lee, Claudia Sippo, Manuel Diaz, and Jennifer Mirish. I've always said to other students, I am very lucky in my dissertation committee because it's always been a very constructive, open environment of discussing where my experiment should go and how I'm progressing. And I've always sincerely appreciated how they've treated me less just like a student or an annoyance or something, a job that needs to be done, and more like a potential future colleague. And I appreciate that. I would like to thank the Beach Lab, Dr. Jordan Beach and his student Melissa, who have graciously shared their technical expertise in confocal microscopy and allowed me to get some of the amazing confocal images that I showed you today. I apologize that through the projector and through the stream, the quality has been lost, but you have to trust me, they are excellent images. The Biochemistry and Molecular Biology Department, all the students there, previous and current, I can't imagine going through all of this graduate stuff without you, and thank you for commiserating with me along the way. And just having someone going through the same thing is pretty nice. Of course, it's headed by the head of my committee, Dr. Mitchell Denny. Loyola University Chicago has always provided a nurturing environment for students. And something I've always said to people visiting the university is that one of the reasons I chose Loyola is I could tell that they're more focused on the growth of the student and what they need as an individual rather than just wringing work out of them. And so I don't think I would be as successful as I am in a different institution. This, of course, includes all of the people, the administration and the staff that ensure that everything works well. Dr. Leanne Cribbs, who has made time for my needs and has discussed many different things with me throughout the years. Thank you to Leanne. Thank you to Lorelei, Sue, and Lynn in the Cancer Center, who do far more work than they get credit for, and everything would fall apart without them. We know that. Thank you to Donna, Anna, Mel, and Margarita in the office, who are always approachable, always make sure things get done and everything's running smoothly. I would like to thank the National Science Foundation, the NSF, who have provided grants that funded all of the research that I showed you, and also funded my living stipend for the last few years, 6, 7 years, however long it's been. And I want to thank all of the thousands or tens of thousands of fruit flies who gave the ultimate sacrifice so that this research could be completed. I thank you. Of course, I need to thank my family and friends, without which I don't think I would be completing this program. You cannot just work constantly. You need to have diversions, you need to have a life outside the lab, you need creative outlets. And everything that I've done with you over my tenure as a graduate student is amazing. I've been able to act as minister officiant at a number of my good friends' weddings. I performed, organized scavenger hunts throughout the year, brought all my friends together, performed in my band, traveled to various places with my friends and family, also produced and directed an independent film that's on YouTube that was mostly done before my graduate school career began. Coaching at a high school speech team that was also mostly done before my graduate school career began. And I can't thank enough my family, who's always been there for me. My mom, Fran, my father, Kevin, my sisters, Claire and Kate, and both of their significant others. For always being there and just always having that family unit, which is absolutely necessary in order to have a basis to get all of this work done. I hope that I can also be that unit to all of them. And of course, I would like to thank Lulu, who is in the room right now, so I'm going to look at her, who is the producer of this video, helping me behind the scenes. But I met Lulu at the beginning of my graduate career, and then we have been together ever since. And we've made a life together, and I can tell you honestly that I would not have been able to complete this program without you. Because having someone else there going through the things, supporting and working with me, I love you, and that is invaluable. Are you crying? Oh, she's crying. No, she's not. But everything we've done together and everything we're going to do together is fantastic. And thank you to all those people, and thank you to all of you at home watching. Whether it's live right now, thank you for dealing with all of the technical difficulties and coming here. Or if you are watching later on, I would love to take any questions right now. So these questions, since there is a delay, of about 30 seconds to a minute. Hopefully people have been writing questions in on the chat, and I'm going to respond to them now. So one of the first questions is, do neurodevelopmental patients with CMI or TRR domain mutations have eye or limb malformations? If so, are patients with CMI or TRR mutations more severely affected? Well, the CMI or TRR orthologs in humans, KMT2C and KMT2D, germline mutation of which causes these developmental disorders which you're referring to, do not have the clear eye and severe eye limb mutations that I've shown you in Drosophila. However, they do have a number of organ malformations, including heart defects and other internal organ defects. Now, one of the reasons that this is true is that if they had more severe mutations, more severe limitations in MLR complex activity in the germline, they would fail to develop completely. Instead, these individuals, just like many other developmental disorders, have just enough mutation, enough reduction in activity in these proteins to survive and develop and grow, but enough to actually affect how they develop. So hopefully, it's been actually demonstrated before that by better understanding the targets and functions of the MLR complexes, it's possible that we can treat these developmental disorders both during development and afterwards. There is some current research into how that's done. The second question is, you mentioned the dysregulation of MLR in cancer. Can you speculate on how your findings in the fruit fly can be translated to better inform therapeutic development in cancer treatment? That's an excellent question. And as I just sort of referred to, there is some research into exactly how we may therapeutically target the defects of MLR complex mutation in developmental disorder. Cancer is a little bit different. Since cancer is a somatic mutation, it can be much more severe loss of function or altered function mutations that cause oncogenesis, since they don't have to develop a whole organism. Instead, it causes malignant cell growth. Now, there is plenty of evidence within these cancers that only one copy of the MLR complex methyltransferases are affected by mutation, suggesting that a wild-type copy is required in order for normal cell function. Now, unfortunately, because the MLR complex is important for regulating a lot of different genes, we cannot dose someone if there was some sort of inhibitor for the MLR complex itself, because that would likely be very lethal. Fortunately, from my research and others, we're demonstrating, elucidating new targets and new interacting partners. And by identifying them, we can then take this basic science to translational cancer research, take a look at cell lines and the like, and see if this is conserved in there, and if so, if by targeting that, we can overcome the detrimental effects of the mutation of MLR complex subunits. And those are the only two questions. So I suppose I'll give everyone 30 seconds right now if they want to type in anything. If you don't, that's fine. Feel free to watch this video again later or the first time, or come to me if you have any questions about my research. Nothing else coming in? Okay, that's fine. Then the last thing that I want to leave you with is this. This over here, this little grain of rice looking thing, is a Drosophila larva. These are, of course, this is on a human hand. It's someone with a very large hand, but still, this is a very, very tiny animal about the size of a grain of rice. These are what I dissected to get the imaginal discs that I showed you. And for a clearer version of that, a clearer picture of that, here are pictures of the eye imaginal discs that I dissected. And next to them are grains of sand. That is how small these organs are. And this is what I had to dissect in order to get these pictures. I just wanted to express that before the end. But afterwards, I want to say thank you all for coming. I now am going to begin my closed defense with my committee.